Bismillahirrahmanirrahim and Assalamu alaikum students. Welcome to our first online class of the course Biosafety and Bioethics, Biot-4803 for the semester Spring 2026, offered in BS Biotechnology 8th semester. To review the overall aspect of this course, we will go through the abstract of this particular chapter and see what main points we are about to cover in the upcoming contents. The advancement in technology is likely to tame several life forms present on Earth. Microorganisms are posing a big challenge due to difficulties encountered to control the disease caused by them. Working with deadly disease causing microorganisms for their characterization, diagnostics, or therapeutics and vaccine development purposes are posing increasingly potential biosafety problems for laboratory workers. Thus an appropriate biosafe working environment may protect workers from laboratory induced infections. Okay, there are different kinds of infections. Okay, there are infections which are caused from animals, which are known as zoonotic infections. Infections that are caused from the source of hospitals, which are nosocomial infections. So there are infections which may be caused from working in a laboratory. तो इस तरीके से हम क्या मेजर्स लें जिसमें हम इन पर्टिकुलर एस्पेक्ट्स को कंट्रोल कर सकते हैं, क्योंकि इन केस अगर कोविड 19 आया था तो although it was a deadly virus, but definitely we need to culture it in the lab to check its genome, its proteome, ताकि उसके अगेंस्ट हम एक इफेक्टिव वैक्सीन मेजर को डेवलप कर सकें. सिमिलरली कोई भी वायरस है, कोई भी बैक्टीरिया है जो पैथोजेनिक है, we need first to grow it in the lab and then analyze it for its physiology, its anatomy, its metabolome, ताकि हम उसको मेन टारगेट पे हिट करें ताकि वो सिंगल हिट में किल हो सके. Biotechnology has the ability to solve the upcoming problems of the world's increasing population. However, there is often reluctance among the public to accept and support biotechnological products in health medicine. Medicine industry or agriculture, there are many safety and ethical issues related to the genetically modified crops and human cloning. Raising transgenic animals and plants faced has fueled ethical concerns, and the scientists have faced a lot of resistance where genetically modified crop plants or reproductive cloning research of human beings is involved. Thus, biosafety and bioethics are continuously being expanded to continue the rationale of ever increasing scientific knowledge in biotechnology that is often in conflict with the long standing social and moral value system of our society. क्या कहते हैं? एक एस्पेक्ट हमारे पास बायोसेफ्टी का हो गया. नाउ बायोएथिक्स, and bioethics is purely related to biotechnology. In biotechnology, we are trying to modify the genomic content of organisms to get a better trait और strain. अब ये जो बेटर का एस्पेक्ट है ये नेचर ने तो हमें नहीं इंट्रोड्यूस करवाया इससे एंड नेचर इज़ द बेस्ट क्रिएशन. ठीक है. तो एक बेस्ट क्रिएशन से भी हमें किसी एस्पेक्ट पे बेटर जाना क्या ये एक क्वेश्चन मार्क आ जाता है जाना चाहिए. तो इस तरह के बहुत सारे कंसर्न्स हैं जिनको एड्रेस करने के लिए बायोएथिक्स की फील्ड डेवलप की गई ताकि ना सिर्फ रिसर्च में हम बायोटेक्नोलॉजी में खुद को एडवांस करें, उसकी परसेप्शन इन द सोसाइटी वो भी उस हवाले से हमारे लिए मीनिंगफुल हो. That advancement in technology is likely to tame several life forms present on Earth. Microorganisms are posing a big challenge due to difficulties in encounter to control the diseases caused by them. Working with deadly disease causing microorganisms for their characterization, diagnostics, or therapeutics and vaccine developments purposes are posing increasingly potential biosafety problems for laboratory workers. Thus, an appropriate biosafety working environment may protect workers from laboratory induced infections. Almost वही चीज़ है जो abstract में हमने देखी है. Biotechnology has the ability to solve the upcoming problems of the world increasing population. However, there is often reluctance among the public to accept and support biotechnological products in medicine industries. There are many safety and ethical issues related to GM crops. However, biotechnological tools have resulted in high yielding crop plants, more nutritive value of food grains, longer shelf life, and resistant to insects and pests. But the public acceptance to these biotechnological products is rather low. For example, GM tools have low support in Europe and India. With the passing years, there is further decline in public support, probably due to media focus and the visible debates on GM crops for fear of the long term effects and unknown risks and environmental safety issues. The controversies from the non government organizations, NGOs, scientists, and media have led to a negative impact on the GM crops. GM crop usage has been controversial, with the fears being expressed on the tomato and many others. GM food again went into controversy due to labeling issues. The BT crops were challenged with the development of resistant insects, so called terminator technology, and a gene use restriction technology GORT met with a lot of resistance due to introduction of negative traits of sterility in the seeds and never came into the market. अच्छा, क्या कहते हैं ओवरऑल जो अप्रोच है दैट इज टू सिक्योर द प्रेजेंस ऑफ जीएम क्रॉप्स इन द मार्केट. बट इसमें कई सारे हर्डल्स आए. अब हर्डल्स ये भी हैं कि एक तो मॉडिफाइड ऑर्गेनिज्म मॉडिफाइड जीनोम है जिसको हमने अपने डेली लाइफ पे यूज़ करना है. सेकेंडली, उन मॉडिफाइड ऑर्गेनिजम्स या मॉडिफाइड क्रॉप्स में देर आर एडिशनल जीन्स. मिसाल के तौर पर हम एंटीबायोटिक रेजिस्टेंट जीन, इंसेक्ट रेजिस्टेंट जीन्स ऐड करते हैं. तो ये चीज़ जो एक किसी भी लाइफ फॉर्म को किल करने के लिए एक एडिशनल फीचर हम इंट्रोड्यूस कर रहे हैं, दैट इज़ कंसिडर्ड एज अ नेगेटिव थिंग. तो क्या ये चीज़ें हम अपनी अतराफ़ में इसको यूज़ कर सकते हैं और हमें नहीं पता कि इसके आने वाले चंद एक सालों में, 10 15 20 सालों में क्या कॉन्सिक्वेंसेस हो सकते हैं। The development of cloned animals and its impact on other wild type animals and the environment also triggered many safety and ethical concerns. Whether or not animals should be used in research, their welfare, sufferings, and well being were debated throughout the world. In India, many plants and animals are linked to religious beliefs and are uh, respected and worshipped for their contribution in the life of human beings. अब वही चीज़ सोसाइटी एग्जाम्पल में इंडिया की इन्होंने बात की है कि वहाँ पे एनिमल्स, कुछ एनिमल्स हैं जिनकी एग्जिस्टेंस इन द सोसाइटी उसकी सेपरेट रिस्पेक्ट होती है। इसके अलावा ये जो जीएम एनिमल्स हैं, जब इनको इंट्रोड्यूस किया जाता है इन वाइल्ड टाइप एनिमल्स यानी कि जो नॉन जीएम है, तो उसमें उनकी इंटरेक्शन, उनके सोशल जो भी कंसिड्रेशंस हैं वो कैसे देखी जाएगी, उन पे भी डिबेट हो चुकी है। Likewise, usage of embryonic stem cells faced controversies and concerns. Protestants agree to have stem cell research under strict public regulations. ये जो क्रिश्चियंस होते हैं उनकी एक कैटेगरी है और उन्होंने फिलहाल स्टेम सेल रिसर्च पे कुछ एडिशनल रिस्ट्रिक्शंस ऐड करके उसको अलाउ किया हुआ है। Many, however, are opposed to embryonic stem cell research as it destroys the embryo. डिस्ट्रॉय से मिलाद के उसके उसको हम अफेक्ट करके ही उसमें हमें एक स्टेम सेल वहाँ से रिकवर करना होता है। The debates and issues surrounding these stem cells are all centered on the potential source of these cells. Embryonic stem cells usage either is prohibited or is under strict public regulation. Somatic stem cells and de-differentiated somatic stem cells can be used for therapy. ठीक है, सोमैटिक मीन्स जो नॉर्मल बॉडी सेल्स है उनको सेल लेके उनसे थेरेपी तो इसको अलाउ किया हुआ है, बट एम्ब्रियोनिक स्टेम सेल्स स्टार्टिंग फ्रॉम द डेवलपिंग एम्ब्रियो, वहाँ से स्टेम सेल्स को वहाँ से स्टेम सेल्स को रिमूव करके उसको प्रोसीड करना है द रिसर्च, ये एक स्टिल कंट्रोवर्शियल क्वेश्चन है। The public support for xenotransplantation with low was low due to safety issues with fear of spread of unknown contaminants. Xenotransplantation, I think, means from other species कोई organ को transplant करना. However, some uh, xenogenic tissue engineered products are now available to treat the dermal wounds and burn patients. The usage of biological weapons, use of living organisms, or its product for human killing in war is presently banned. Biosafety and biorisk. The biotechnology has tremendously evolved, including the technical aspects of medical sciences like diagnostics and therapeutics. With all these, we are also witnessing a rapid and dangerous adaptations in microorganisms, especially for developing resistance to antibiotics. ठीक है, ये कि माइक्रो पे एंटीबायोटिक रेजिस्टेंस को एक तो खुद इंट्रोड्यूस किया जाता है, बाकी चूंकि इतलाफ वजूद देर आर सो मैनी chemicals that are being trying to किल द बैक्टीरिया, तो वो नेचुरली अपने बॉडी में, सॉरी, अपने सेल में स्टार्ट डेवलपिंग enzymes which are now able to degrade those antibiotics. Microbial Microbial pathogens are causative agents of many diseases, and due to uh, mutation in their genes, they have developed a multiple drug resistance. It is gradually becoming difficult to control those multi-drug resistant infections, pathogenic microorganisms. To search for solutions to overcome the multi-drug resistance of these pathogenic agents, several scientists and medical workers are heading handling these pathogens. This poses enormous biohazards and raises serious biosafety issues. That is, the scientific practices, methods, and use of appropriate equipment in biosafety environment. ठीक है, जो साइंटिस्ट, जो वर्कर्स इन मल्टी ड्रग रेजिस्टेंट बैक्टीरिया को हैंडल कर रहे हैं, उन पे स्टडी कर रहे हैं, उनको एक्सपेरिमेंटल कल्चरिंग कर रहे हैं, तो उनके लिए एक बहुत ही सीरियस बायोसेफ्टी हैजर्ड कंसर्न है. दे नीड टू डेवलप सम मेजर्स जिससे उन रिसर्चर्स, उन वर्कर्स को लेबोरेटरी में उनको खुद से इन्फेक्शन ना हो सके. The biosafety aspects have become very important in various conditions and require many precautions in healthcare systems, as hospitals, diagnostic laboratories, animal care systems, biological laboratories, and so on. The precautions which can be taken to reduce or nullify the risk associated with samples by commonly monitoring and recognizing potential hazards, their risk assessment, and preventive measures to avoid exposure which might result in infection. Individual worker should be appropriately trained and must understand the conditions like containment, which is the conditions where infection agent can be safely manipulated, and good laboratory practices which can minimize exposure to pathogens. ठीक है, वो मेजर्स हमें लेने पड़ेंगे जिसकी मदद से हम इन तमाम जो कंसर्न्स हैं, जो हैजर्ड्स हैं, उनको मैक्सिमली रिड्यूस कर सकें. Biorisk is the likelihood of the occurrence of an adverse event. Thus, biorisk is the likelihood of the occurrence of serious infection due to exposure or to pathogenic microorganisms or biohazards. Upon exposure, there may be mild to severe infections, allergies, and other clinical problems associated with the pathogen. The biorisk can be managed by risk assessment, effective biosafety measures, and bio containment. The first report on laboratory associated infections was published at the beginning of the 20th century, 1900s में कहीं. However, 4079 infections associated with laboratory were reported with 168 deaths somewhere between 1930 and 1978. The major pathogenic agents responsible for these infections were Brucella species, many technical names, blastomyces, dermatitis, chlamydia, coccidioides, coxella, hepatitis B virus, Salmonella typhi, Francisella Mycobacterium tuberculosis, and Venezuelan equine virus. After this 1978 report, many more laboratories associated infections and deaths were reported. In these cases, also the infectious agents were uh, different other bacterial and viral species, Shigella, tuberculosis, Salmonella species, hepatitis C virus. Assessment of the risk. The processes used to risk assessment are identification of the hazardous properties of a familiar infectious agent or material. The activity is responsible for pathogen exposure to the person. The chances of the exposure becoming a lab associated infection and the ultimate consequences of infection. ये हमारे पास रिस्क असेसमेंट के चार स्टेप्स हैं और इसमें हम इंडिविजुअल लेवल पे उस पैथोजन को स्टडी करते हैं और प्लस उस पैथोजन से रिलेटेड माइक्रो ऑर्गेनिजम्स को भी स्टडी करते हैं टू परफॉर्म कंपैरेटिव एनालिसिस एंड टू चेक कि किस तरीके से ये जो पैथोजन है दैट इज़ डेडली फॉर द ह्यूमंस और जो रिलेटेड स्पीशीज हैं दैट आर नॉन डेडली फॉर द ह्यूमंस. Risk assessment needs a very alert judgments. If risks are underestimated, they may result in serious and adverse consequences. Overestimation of risks can result in undue protective measures, which may be a burden for the laboratory. ठीक है ना ही हमें अंडर एस्टिमेशन करनी है ना ही ओवर एस्टिमेशन करनी है बट एक स्टैंडर्ड वे में हमें एनालिसिस करके असेसमेंट करके उसके रिस्क को नोट डाउन करना होता है. Assessment of risk also needs to evaluate the factors responsible for infections, whether it is due to hazardous agents or due to hazards of laboratory procedure or the working attitude of the laboratory staff. ठीक है ये सिर्फ पैथोजेंस की वजह से नहीं डिफरेंट अदर इन्वायरमेंटल फैक्टर्स मशीन एरर ह्यूमन एरर इस वजह से भी ये रिस्क जनरेट हो सकते हैं. The staff capability can be improved by appropriate training and enculturing good laboratory practices with training of accidental spoilage and inoculations. ठीक है जो स्टाफ है उसकी ट्रेनिंग अपनी जगह पे बहुत ही ज़रूरी होती है टू get overcome by this biorisks and biohazards. ठीक है, बायोहैज़र्ड्स को हम इंशाल्लाह कल वाली क्लास से ही स्टार्ट करें, कल नहीं जो हमारी नेक्स्ट क्लास होगी। होपफुली नेक्स्ट क्लास हमारी टाइम टेबल में कब है? इट्स ऑन मंडे 12:30 to 1:30। ठीक है,